Welcome to Lecture 10.2, in which we will discuss non-Mendelian inheritance. This image shows William Bateson, a biologist who rediscovered Mendel's work and helped expand it in the early 1900s. He was a bit unique in that he ran a research group mostly composed of women, including his wife and sister-in-law, who are sitting next to him here, attending a conference on genetics. His sister-in-law, Florence Durham, performed experiments that contributed to our understanding of epistasis, which we will talk about later in this video. Our learning objectives for this video are to identify non-Mendelian inheritance patterns, specifically polygenic traits, pleiotropy, multiple alleles, epistasis, incomplete dominance, codominance, and sex-linked traits. We will also talk about what X inactivation is and why it's important. One of the good things about pea plants is that each of their traits were linked to a single gene, making it easier to isolate the inheritance of a single characteristic. However, many characteristics are determined by multiple genes. For example, eye color is mostly determined by two genes, commonly thought of as the genes that determine whether an individual has brown, blue, or green eyes. However, there are many more possible eye colors, and at least 16 contributing genes have been identified. On the other end, we have pleiotropy, which is when one gene manifests as multiple phenotypes. This is very common with single genes that cause diseases. For example, cystic fibrosis is caused by a mutation in a gene called CFTR. This gene creates a chloride channel that is found in many different types of cells. Since so many cells are affected if this gene is mutated and the chloride channel isn't working, many parts of the body are affected, including the nose, lungs, heart, and digestive system. Genes also generally have more than just two alleles. For example, blood type has 3 alleles. This is generally designated as an uppercase I with an A or a B, or a lowercase I. IA and IB are codominant, which we will discuss a bit later, and both are dominant over lowercase I. Another unique case is epistasis, which is when one gene affects how another gene is expressed. Generally, this is because certain alleles for one of the genes are needed for the other gene to work properly. The classic example of this is mouse coat color, and many of these breeding experiments were done by Florence Durham, sitting on the right in the title slide. In this example, coat color is determined by two genes. One gene, designated as C, determines whether the mouse can produce the pigments needed for there to be a coat color. Having a coat color is dominant over not having a coat color. The other gene, designated as A, determines whether the mouse has an agouti coat color, which is the yellow color or the black coat color. In this case, agouti is dominant over black. The gene that allows for pigmentation, C, is needed for the mouse to have a coat color. So if the mouse is recessive for coat color and has no pigmentation, it would have a white coat regardless of what the genotype for the other gene is. This results in a slightly different distribution from a dihybrid heterozygous cross, in which 9 out of 16, as usual, have the dominant traits in the form of agouti coat color, 3 out of 16 are recessive for gene A, but dominant for having coat color, and so they have a black coat, and 4 out of 16 have a white coat because they are recessive for having coat color, even though they have at least one dominant allele for the agouti gene. There are also a few exceptions to dominance. Incomplete dominance is an example in which the heterozygous phenotype is an intermediate of the two parents phenotype, which means that instead of one allele being dominant over the other, neither allele is dominant, and both are partially expressed. A common example is a snapdragon. Here we have allele C for color, with R designating a flower with a red color and W designating a flower with a white color. When crossed together, they produce offspring that are all heterozygous, and they have pink flowers instead of all red or all white. If two heterozygous offspring are crossed together, we would still get the expected ratios of 1/4 being homozygous for having alleles coding for red, another quarter homozygous for having alleles coding for white, and half that are heterozygous. This results in 1/4 being red, 1/4 being white, and half being pink. It is also possible to have codominance, which is when both alleles are fully expressed together, rather than partially expressed. The most common example is blood type, which we discussed earlier. As mentioned, IA and IB are codominant and dominant over lowercase I. These manifest as type A, type B, or type A and type B surface markers on a red blood cell, or no surface markers at all for someone who is type O. If someone heterozygous for blood type A had children with someone heterozygous for blood type B, it would be possible for them to have children who are type AB by receiving the IA allele from one parent and the IB allele from the other, or blood type A or B due to receiving IA or IB from one parent and the lowercase I from the other, or blood type O due to receiving recessive alleles from both parents. Genes can, of course, also be found on sex chromosomes. Remember that a biological female would have 2 X chromosomes, and a biological male would have 1 X and 1 Y chromosome. Both chromosomes have genes, and these genes code for sex-linked traits. The X chromosome is larger, and so most sex-linked traits are actually also X-linked, meaning the genes that code for these traits are found on the X chromosome, but not the Y chromosome. An example of a sex-linked trait is eye color in fruit flies. The dominant phenotype is red eyes. We use X and Y to represent the X and Y chromosomes, and the uppercase R next to the X represents the allele for red eyes. The lowercase r represents the allele for white eyes, which is recessive. The male has 1 X chromosome and 1 Y chromosome, with the dominant allele on the X chromosome, so he has red eyes. The female has 2 X chromosomes, both with the recessive allele, so she has white eyes. In the Punnett square, the male's X chromosome goes in the first row, and the Y chromosome goes in the second row. Likewise, one of the female's X chromosomes goes in the first column, and the other goes in the second. The male parent is the one who determines the biological sex of the offspring, since only he can give the X or Y chromosome, and the female parent can only give X chromosomes. Offspring who receive the X chromosome from the male are female, and in this case they get an X chromosome with the dominant allele from the father, and an X chromosome with the recessive allele from the mother. Since they have 1 dominant allele, they have red eyes, but they are carriers for the recessive allele. If they were to go on to breed with another male, they have a 50% chance of passing on the X chromosome with the recessive allele, which means that there is a 50% chance that the males in the next generation would have white eyes. The males in this generation receive the Y chromosome from the father, and so the X chromosome could only come from the mother. Since the mother only has X chromosomes with the recessive allele, the sons end up having 1 X chromosome with a recessive allele and 1 Y chromosome. There is no second allele, so the sons can only have white eyes. There are a few X-linked traits in humans as well, with the most common example being color blindness. It seems that humans only need 1 X chromosome to survive, but how is this the case? Why are there no detrimental effects of just having 1, or of having more than 1? The answer is something called X inactivation. This is a process in which an X chromosome in each cell gets inactivated, and therefore its genes aren't expressed. This only occurs when there are at least 2 X chromosomes, so the X chromosome in males would not get inactivated. The X chromosome that gets inactivated is generally random. This is actually what creates the pattern on calico cats. The X chromosome that is not inactivated determines whether the fur is orange or black in an area. In this video, we learned about various non-Mendelian inheritance patterns. Polygenic traits are traits encoded by multiple genes. Pleiotropy is when a single gene instead affects multiple traits. Multiple alleles is when a gene has more than just 2 alleles. Epistasis is when one gene affects the expression of another gene. Incomplete dominance is when 2 traits are expressed equally, but not completely, creating offspring that looks like an intermediate between the 2 parents. While codominance is when the 2 traits are both fully expressed at the same time. Sex-linked traits are determined by genes located on sex chromosomes. Since females have 2 X chromosomes, X inactivation occurs to ensure that there is equal expression of X chromosome genes, regardless of sex. Thanks for watching.